Hello students, myself Dr. Swati Talele, Associate Professor, Pharmaceutical Quality Assurance Department. Here I am going to deliver the lecture on Novel Drug Delivery System for the class final year B Pharm on the topic microencapsulation. So let us see the course outcome for this subject. Learner will be able to relate the concept of novel drug delivery system, outline the various approaches for the development of novel drug delivery system, identify the criteria for the selection of excipient in formulation, then analyze the factors that contribute to the various evaluation parameters in the formulation development, compare underlying techniques used in the novel drug delivery system, then elaborate on current advances and emerging technology in the novel drug delivery system. Now, what is the learning outcome of the topic microencapsulation? The student understand the fundamental concept, know about the classification of microencapsulation, know about microencapsulation technique, understand kinetics of release from the microcapsule, learn about evaluation properties of microencapsule. Now, content of the presentation: introduction, classification, advantage, disadvantage, application, fundamental consideration, microencapsulation technique, kinetics of drug release from the microcapsule, then evaluation of microencapsule, marketed preparation, references, and some of the MCQs for the student. Now, let us see about what do you mean by microencapsulation. So microencapsulation, it is the novel technology. It's the process or technique by which relatively thin coating can be applied to the small solid particle or the droplets of liquid or the liquid dispersion. Now this product whichever to be obtained, this is called as a microparticle or the macrocapsule. If you see the diagrammatic representation, the inner, it shows that here the core drug which is called as the nucleus or the field, which is most probably 10 to 90%, and the outermost layer which is called as a coating layer, capsule wall or the capsule shell. This microencapsule, it has the particle size in between 1 micrometer to that of the 5000 micrometer. Whenever the particles are smaller than 1 micrometer, these are to be referred as nanocapsule or the nanoparticle. Now let's see about the classification of microencapsule. So depending on the size or the manufacturing process, various types of microencapsulant it is to be get obtained. So first let us see that is monomolecular spherical, where the innermost is the nucleus and the outermost is the coating. Second one is the multinuclear spherical. The same concept again it is to be there, but instead of the one more nuclear formation it is to be there. Next to that of the multinuclear irregular. In case of the spherical shape, it is to be replaced by the irregular shape. Next one is that multinuclear irregular cluster. Cluster formation is there. Next, encapsulated microcapsule. Initially, microcapsule it is to be encapsulated. Again, that same microcapsule it is to be one more encapsulation, that is the coating it is to be there, that is called as an encapsulated microcapsule. Next to that of the dual wall microcapsule. So these are about the classification. So let's see about what are the advantage and the disadvantage of the microencapsulation. As I already discussed, microencapsulation, this is the one of the new dosage form novel drug technology. Now this will be convert most probably liquids to that of the solid. Example is that here iprazine. Another one is that here this microencapsulation, there is the alternation in colloidal as well as the surface property. This will provides the environmental control to that of the drug which will sustain to that of the environment. This microencapsulation, it has the ability to control the release of the drug. Next, it has some disadvantage, that is many times those formulator or the manufacturer they face the some formulation difficulty. No single microencapsulation process it is to be applicable to all the core material, that is all the API. Many times we observe that incomplete or the discontinuous coating it might be get occur. Sometimes failure to produce the predicted drug release. Some of the economical limitations again it is to be there. Cost is always more than that of the conventional tablet as well as the capsule. Now, what are the applications of the microencapsulation? Microencapsulation, as it provides the inner core and outside is the coating material, so it can be useful in insecticide, that it reduces the hazards. Then again, it will reduce the possibility of sensitization just because of the coating. Example is the ampicillin trihydrate. Again, it will be more advantageous to that of the hygroscopic material, for example sodium chloride. Next, it will improve the flow properties of vitamins, for example, riboflavin, then niacinamide, then thiamine, again it is there. Then it will provide the protections to that of the core material from the environmental conditions, so that is to be more applicable for the microencapsulation. It also enhances the stability of the fat soluble vitamins. After that, here if any incompatibility it is to be get occur in between the drug, this can be get prevented with the help of microencapsulation, and the best example is the aspirin and the chlorpheniramine maleate. Another one is that they have the property to mask the unpleasant taste of the drug. Acetaminophen, that is the example. If any gastric irritation, if it is to be get observed, for example, aspirin and potassium chloride, so that gastric irritation again it can be get reduces, then it will provide the sustained release type of the dosage form. Now, what are the fundamental consideration of microencapsulation? So, in fundamental consideration, mostly microencapsulation, it consists of core material, coating material, as well as the vehicle. So, core material, it can be the solid, it can be the liquid. Whereas the coating material, we can be used as a water soluble resin, water insoluble resin, then waxes and the lipids also there, and the enteric resins. And number of examples here I put it. Now, what are the vehicles it can be get utilized in microencapsulation? In microencapsulation, we can put the aqueous as well as a non-aqueous vehicle. Now, again in fundamental consideration, what the core material and what is the coating material. Core material, it is defined as the specific material to be get coated. So specific coating material, that core material, it can be the liquid or it can be the solid as per the previous discussion. Now the composition of core material, it is nothing but here your drug or the active constituent, then additives like diluent, stabilizer, again it is to be there. So this some of the release rate enhancer or the retardants again we can be used. Now as per the diagrammatic presentation, we already seen the discussion. Core material, that is the inner material, and the coating material is the outside material. So coating material properties, it's that it should form the thin film cohesive with the core material. Then it will be or it should be chemically compatible, non-reacting with the core material. Then this coating material again it will provide some desired coating property, like the strength again it should be there, flexibility, impermeability, and the stability again it should be there. Now, various microencapsulation techniques are air suspension technique, conservation phase separation technique, then spray drying, spray congealing method, multi-orifice centrifugal process, pan coating, solvent evaporation, as well as the polymerization. So, very first one, air suspension technique. So as per the diagrammatic presentation here, this air suspension technique, we can form the part approximate size range 35 to 5000 micron, and this is more applicable for the solid material. So this microencapsulation initially invented by Professor Dalla E. Worcester, and that's why this process it is called as the Worcester process. So basic principle of this air suspension technique is that here the solid core material it is to be just get passes in a supporting air stream, that is called as what, fluidized fluidize or the suspension of core material in air, and then here the spraying of the coating solution on air suspended particle, it is to be get occur. So as the core material fluidize in a air stream in this method, and then coating it is to be get occur, so this method it is called as the fluidized bed coating. So as per the diagrammatic presentation, the coating material it might be get applied either at the top or either at the bottom as shown in the diagrammatic presentation. Next technique, conservation phase separation. Conservation phase separation, these are characterized by the three steps. Initially there should be the formation of three immiscible phases, then there will be the deposition of coating material. After the deposition of coating material, rigidization of the coating it should get occur, and whenever the coating material phase form, then number of methods these are to be get utilized. So either the change in temperature, either the addition of incompatible polymer, or either the solvent solvent and non-solvent addition by interaction, again it is to be there, by solvent addition, polymer interaction, again it should be there. So let's see about the conservation phase separation, that is change in temperature. So in this change in temperature method, if we take the core material, that is the N-acetyl para-aminophenol, the polymer it use as ethyl cellulose, solvent it use as a cyclohexane. Why we are using the ethyl cellulose? Because ethyl cellulose is insoluble in cyclohexane at the room temperature, but it is soluble at the elevated temperature. So initially we will take here the 2% weight by weight ethyl cellulose in cyclohexane. After that, heat this mixture to the boiling point. So why the heating is essential? Because it forms the homogeneous polymer solution. After that, add the core material with stirring. Now cool the mixture at room temperature with stirring. Immediately there will be the formation of microcapsule. So filter it, dry it. So this is about the change in temperature by conservation phase separation. Now here there will be the formation of what here, this phase diagram, again it's there. So in this phase diagram, the X it acts as what, single phase homogeneous solution at all the points or above the phase boundary, or this is also the binodal curve formation, again it is there, and that binodal curve formation is FEG. Now, as the temperature decreases from the point A along the arrow line AEB, the phase boundary cross at the point E and the two phases region it will get enter. Now, B again it indicates the segmented tie line, that is the vehicle phase. C it shows coexisting phase, and D is exists, that here the concentration of polymer solvent mixture. Now let's see about the second method, incompatible polymer addition. Core material crystalline methylene blue hydrochloride, let's take. Then polymer ethyl cellulose, solvent is toluene. And incompatible polymer, here we are adding polybutadiene, that is in liquid stage. So initially dissolve ethyl cellulose in toluene to produce 2% weight by volume solution. Then disperse here the core material crystalline methylene blue with stirring. Then slowly add this polybutadiene which is in liquid form. So solidification it get occur by addition of the hexane. Once you add the hexane, there will be the formation of microcapsule. So take that microcapsule, filter it and dry it. This is the second method. Third method is non-solvent addition. Here in non-solvent addition, we are adding isopropyl ether. Core material it is selected as methyl scopolamine hydrobromide. Coating polymer we are adding cellulose acetate butyrate. Solvent we are adding methyl ethyl ketone. So let's take the coating material methyl scopolamine. This is to be added in a solvent methyl ethyl ketone. Then add the core material, that is methyl scopolamine hydrobromide. Then it should be get dispersed with the stirring. Then heat the mixture at 55 degrees centigrade. Then add the non-solvent. After addition of non-solvent here, let's cool the mixture. After the cooling the mixture at the room temperature, there will be the formation of microcapsule. Next method is the salt addition. So in this case, we take the core material as a oil soluble vitamins and the oil as a corn oil, aqueous phase as a water, polymer we use in this case as a gelatin. So dissolve the core material oil soluble vitamins in a corn oil, then emulsify 10% gelatin solution at the 55 degrees centigrade. Then we have to add the salt, that is 20% sodium sulfate solution. Once you add the 20% sodium sulfate solution, we have to again go for the rigidization process by addition of 7% sodium sulfate solution, but maintain the temperature as a 19 degrees centigrade. So once you maintain the temperature at 19 degrees centigrade, now you will get some sort of microcapsule. So filter it, wash it with water. Next method, polymer-polymer interaction. So this polymer-polymer interaction, this is to be based on ability of the cationic anionic water soluble polymer to interact in water to form a liquid polymer rich phase complex coservate. So core material in this case we are using methyl salicylate, positively charged polymer gelatin, negatively charged polymer as a gum arabic. So there will be the formation of, that is the microcapsule, by the polymer-polymer interaction, that is positively charged polymer as a gelatin and negatively charged polymer as a gum arabic. So initially the core material methyl salicylate, it is added in aqueous gelatin solution, then add aqueous gelatin solution which carries the positive charge polymer, then add the gum arabic solution which has the charge as a negative, adjust the pH in between 4 to 4.5, cool to room temperature. Once you cool to the room temperature, there will be the formation of microcapsule with the rubbery shell. Cool to 70 degrees centigrade, then treat the solution with the glutaraldehyde as a cross-linking agent. Once we add the glutaraldehyde, there will be the formation of microcapsule. Now, this is the diagrammatic presentation of polymer-polymer interaction or the complex conservation as I discussed earlier. Next technique, spray drying and the spray congealing. So the spray drying, it is the process where the coating solidification, it is most probably affected by the rapid evaporation of the solvent in which the coating material is dissolved. Whereas the spray congealing, this is the coating solidification, and this is to be affected by thermally congealing the molten coating material. So in this spray drying method, we are getting the size of the particle 5 to 600 micron, and this is applicable for both solid as well as the liquid process. So in this process, if we see the diagrammatic presentation, it has, that is the construction, initially pump the drug polymer solution, it is to be just get and enter into the drying chamber. After that, here it has the air heater, then again have the some cyclone separator for the separation of the particle according to that of the particle size. After that, here again it has some filter, then condenser, again it is to be get attached. So this spray drying, this technique, it can be applicable for both the solid as well as the liquid, and it has a wide application in the pharmaceutical industry. Multi-orifice centrifugation technique. So this multi-orifice centrifugation technique, this technique initially developed by the Southwest Research Institute, and in this technique we generally core material inlet, then core material coating material, there should be, that is the core material inlet, it will be passed through that of the one of the disc as well as the one of the rod, again it is to be given. So on that, here core material, it is to be just taken. After that, the coating material, again it will be passed over this particular, now this is to be totally get operated under the outer rotating disc, and there will be again some flow of the core inlet as well as the coating inlet. It should be just get maintained, so that here the proper coating material it will be get obtained. But multi orifice centrifugation, the name itself indicate that here some centrifugal force it is to be applied on the core material by the envelope microencapsulated membrane. So in this technique we can say that here ki this is relatively simple technique, high production efficiency technique again it is there, then cost effective and the rapid technique again it's there. And but in this case some of the factors we have to take into consideration whenever we are going to form this microcapsule by with the help of this technique. That is cylinder rotation speed we have to maintain, coating core material flow rate again it is to be maintained, viscosity, surface tension, the concentration of the core material again it should be get maintained. Next technique is the pan coating. So this is mostly relatively used for the large particle. This is used for the preparation of the control release beads. Then again this will be form the spherical substrate. Then again in this case we can apply the coating material as a solution or as an atomized spray to the top the solid core material which is to be present in a coating pan. Now the approximate particles which is to be get formed, that is 600 to 5000 micrometer, and this technique it will be used only for the solid. So this technique it is totally based on what here some air atomization and on the spray nozzle. Next is the solvent evaporation technique. So solvent evaporation technique it is based on that the core material initially dissolve in or the disperse in a coating polymer solution. Then with agitation we have to add the liquid manufacturing vehicle phase. Heat if it is necessary, then there will be the evaporation of polymer solvent and then formation of microcapsule again it is to be there. So approximate size it get formed, that is 5 to 5000 micrometer. It can be applicable for the solid as well as the liquid. Next, that is the polymerization technique. So this technique it is the new and microencapsulation method, where here polymerization techniques most probably get utilized to form the protective microcapsule. So this method it will involves the reaction of monomeric unit which is located at the interface, which is to be existing between a core material substance and the continuous phase in which the core material is dispersed. The core material supporting phase usually a liquid or the gas, and therefore polymerization reaction it get occur at liquid liquid, liquid gas, solid liquid or the solid gas interface. For example, this polymerization technique it get utilized in the formation of polyamide, that is the nylon. Polymeric reaction is occurring at the liquid liquid interface. Now this is about the interfacial polymerization and in situ polymerization difference. Interfacial polymerization it forms the shell over the droplet or the particle by the reacting monomer. Then the multifunctional monomer it is dissolved in the liquid core material and then it is dispersed in the aqueous phase containing the dispersing agent. The co-reactant amide is added, which will result in the rapid polymerization at the interface and the microcapsule it get generated. Whereas in situ polymerization, this technique somehow it is similar to that of the interfacial polymerization, that will get shale formed by the polymerization. But in this technique, we are not using any reacting agent which is to be added in a core material. So polymerization itself it starts in a continuous phase and it deposited at the interface. So once the time it will get progress, the pre-polymer grows in size. Next, what are the kinetics of the drug release from the microcapsule? So major mechanism of drug release from the microcapsule, it includes diffusion, dissolution, osmosis as well as the erosion. So most common mechanism we observed for the drug release is nothing but here the diffusion. Next, that is the dissolution. So initially we have seen that here ki diffusion technique is most probably get utilized. So in dissolution, the release rate of the drug from the microcapsule, this is totally depends on the dissolution rate of polymer coat and the solubility of the coat in the dissolution media. So generally solubility in the dissolution fluid and the thickness of the coat, it will influences the release rate. Osmosis is again another process. So essential requirement of osmosis is the semi-permeable membrane. So once the process is get progress, osmotic pressure is get created in between outside and the inside membrane of the microcapsule, and which will leads toward the release of the drug through the small pore. Another technique is the erosion technique. So erosion of the coat, it is totally occur due to the pH, enzymatic hydrolysis, and it causes the release of the drug with certain core material like beeswax, sterile alcohol, glyceryl monostearate. So this drug release from the microencapsulation, this has become complicated because of the diversity formation or the diversity in the form of microcapsule with the shape, size, arrangement of the coat as well as the coating material. Now let's see about after the formation of microcapsule, what will be the evaluation parameters of the microcapsule. Following parameters, these are to be get carried. Morphology, drug content, encapsulation efficiency, percent yield, bulk density, angle of repose, particle size determination, in vitro dissolution, drug release stability study, and the assay. So in morphology, optical microscopy is most probably get utilized to determine the shape of microcapsule. Shape and surface morphology, again it can be get carried out by the same. So it totally gives the idea about that is surface before the coating, after the coating. It also determines the uniformity and the nature of coating shape. Next evaluation parameter is the drug content. So generally microcapsule, it is selected from the various batches randomly, and specific quantity of the microcapsule, these are taken, then mechanically powdered. These are to be dissolved in a dissolution media of the specific quantity, then filtered through the Whatman filter paper, then prepare this, now take this solution, and this is to be again analyzed by the sophisticated technique, that is nothing but here the UV visible spectroscopy. And the formula for that one again, you can be it can be get utilized, that is the drug release in is that is concentration into 1000 dilution factor divided by the weight of that is the particular type of the drug. Encapsulation efficiency, again it can be determined by actual drug content and the theoretical drug content into 100 percent yield. This can be get calculated by the formula, that is the weight of microcapsule as a m0, it is the theoretical weight of the drug plus your polymer. Bulk density, this can be determined by the three-tap method, that is the mass of the powder divided by the bulk powder. So weight of the sample divided by the final volume after the tapping. Angle of repose, this is to be determined by the funnel method, and the formula tan theta is equal to h by r. H is the height of the pile, r is the radius. Now particle size determination. So size distribution, again it plays a very important role in determining the release characteristics of the microcapsule. So it will determine the variation in the coating. So size of the coated particle is one of the technique to check batch to batch variation. So particle size, as we discussed, this will be determined either with the help of the optical microscopy or the same technique it can be get utilized. In vitro dissolution drug release, it can be determined by using the USP dissolution method, that is as per the pharmacopoeia what the dissolution media it is to be given. Next, stability study. Stability study, it is to be either conducted for the short term or the long term, or the as per the ICH guideline. The parameters this will be considered are, that is temperature as well as the relative humidity. Now the assay. The assay of the microcapsule, this is to be get carried out by dissolving the specific weight of microcapsule into methanol or any other suitable solvent. Then filter through the 0.45 micron, and then appropriate method it can be utilized for the estimation of the drug content. So assay it is to be carried out on each batch in an at least 6 times in order to check batch to batch variation. Next, these are some of the marketed preparation: cephalaxine, cefadroxil, that is for the bacterial infection, then Zotrin tablet, that is aspirin for relieving arthritis symptoms, then again Lupron Depot injection, then another one is that Lomitrigine, this is another example, then vitamin C microcapsule. Now some of the properties of microencapsulated core material. So let's see one or two example. If the core material is the acetaminophen, the properties is the slightly water soluble. So purpose of microencapsulation is test masking, and the final product is the tablet. So here we can take acetaminophen, activated charcoal, then aspirin, then islets of Langerhans, isosorbide dinitrate, liquid crystal, menthol, methyl salicylate camphor mixture, potassium chloride, urease, vitamins A palmitate. That is the purpose of encapsulation. It is already quoted here. So these are the references. And at last, some of the MCQs on microencapsulation for the student. So thank you all of you.